Welcome to ECCO 2024, where we present a poster comparing the clinical performance of 4 different fecal calprotectin assays. Endoscopy is the gold standard to differentiate between IBS and IBD, and also to follow up diagnosed IBD patients. But it has been shown that fecal calprotectin is a very good surrogate marker for mucosal inflammation, and it's being used to help in the aid of disease diagnosis and also disease course follow-up. There is no international standard for calprotectin, so all the manufacturers are using internal standards to make sure that all their assays compare well with each other. Bühlmann has 4 different assays: a standard ELISA assay, a high throughput to the metric assay, and a lateral flow test that can be analyzed with a tabletop lateral reader and as a home test with smartphones and the backbone of the smartphone server. So we took 128 samples from an FDA submission study where patients presented with signs and symptoms of mucosal inflammation and then underwent endoscopic assessment. We measured all the samples once with all 4 methods, and then we calculated a ROC curve. And as you can see here, the area under the curve for all the assays are very similar, showing a similar clinical performance. We also looked at the sensitivity and specificity of the cutoffs that Bühlmann recommends, so that for diagnosis 80 and 160 micrograms per gram, and for disease follow-up of diagnosed patients 100 and 300 micrograms per gram. And as you can see, all sensitivity and specificity at all cutoffs are very comparable between the different assays. We also calculated estimate % agreement for positive and negative agreement between 2 smartphones of the home test and the metric laboratory assay, as well as supportive care lateral flow assay. And also here, the PPAs and the NPAs are highly comparable. So in conclusion, we can say all the assays measure very similarly. The clinical performance is very similar. So then the assays can be used interchangeably, and the clinical care team can decide on what combination of assays fits best their clinical needs. Thank you very much.